Good morning, everyone. Welcome to Udan Career Institute. In today's class, we will continue with chapter 2, that is reproduction in angiospermic plants. Now, students, in the last class, we have gone with the process of double fertilization. We have completed the double fertilization. In this particular topic, we have studied that two different areas where the fertilization takes place, where the center part of the cell and the egg cell which was present at the micropylar end. So two types of fusions were taking place. One was syngamy and the second one was triple fusion, where syngamy results in the formation of a zygote, and a triple fusion it leads to the formation of primary endospermic cell, and this primary endospermic cell it leads to the development of the endosperm. Now in today's class we will continue with another topic, that is post fertilization structure and the events which are involved with post fertilization. Now post fertilization events, they include all those events which takes place after the process of fertilization of male gamete and the egg cell. So after the formation of the after the fertilization, the zygote formation takes place, and this zygote it further leads to the development of embryo. But along with embryo, endosperm is also formed. So post fertilization event it includes development of endosperm as well as development of embryo. Development of endosperm it takes place from PEC, that is primary endospermic cell, and development of embryo it takes place from the zygote. Also there will be maturation of ovule into seed, and ovary will mature into fruit. So these are the four major events which takes place in case of post fertilization. Now we'll go with the first one, the development of the endosperm. Now endosperm development it takes place before the embryo development starts. Now the primary endospermic cell which has been formed by triple fusion, it divides repeatedly and it forms triploid endosperm tissue. So this tissue, that is the triploid endosperm tissue, it contains cells, and these cells they are filled with reserve food materials, and they provide nourishment to the developing embryo, and that is why the endosperm development it precedes the embryo development, so that sufficient amount of nutrition can be provided to the developing embryo. Now, in case of most common type of endosperm development, the pen, that is primary endospermic nucleus, it will undergo successive nuclear division and it gives rise to free nuclei. At this particular stage of the endosperm development, there will be various free nuclei which will be formed, and hence it is known as free nuclear endosperm. Now, subsequently, after the formation of free nuclei, the cell wall formation takes place and the endosperm will become cellular in nature. The coconut water, which is present in coconut, it is free nuclear endosperm. That is, it is made up of thousands of nuclei, and the surrounding white kernel, it is the cellular endosperm. So basically these are the two major types of endosperm which are found mostly in angiosperms, that is the free nuclear endosperm and the cellular endosperm. The surrounding white kernel, which is the cellular endosperm, it is mostly the eatable portion or edible portion of the coconut. Now endosperm, it is either consumed totally during the development of the embryo, which is seen in case of pea, groundnut and beans before the seed maturation takes place, or this endosperm may persist in mature seeds, example castor and coconut, and it can be used up during the seed germination. So there will be two types of seed which are found depending upon the presence of endosperm or depending upon the absence of endosperm. So the seeds in which the endosperm it is completely consumed and in which the endosperm is not found, those kind of seeds are basically known as non-albuminous seeds, whereas the seeds in which the endosperm persists, such kind of seeds are known as albuminous seeds. Now, if you split open some seeds of castor, pea, beans, groundnut, fruit of coconut, and look for the endosperm in each case, and we can see that in many of the case endosperm is persistent. Now, endosperm basically it is not found in case of non-endospermic seeds or non-albuminous seeds. Now, some examples of non-albuminous seeds, they are beans, pea and groundnut, whereas the examples of albuminous seeds, they are wheat, maize, castor, coconut, sunflower. So, in case of wheat, maize, castor, coconut, sunflower, residual endosperm is present, whereas in case of beans, pea, groundnut, the endosperm is used up and it is not present. Now, after the completion of the development of the endosperm, we move forward towards the second event, which happens in case of post fertilization, that is, development of embryo. Now, as there are two types of plants which are mainly found, they are dicots and monocotyledon plants. First, we'll go with development of embryo in case of dicotyledonous plant. Now, embryo it develops at the micropylar end of the embryo sac where the zygote is situated. So, this is the location where the embryo develops. So, embryo is developing at the micropylar end because zygote is situated at the micropylar end. Now, mostly zygote divides only when certain amount of endosperm is formed. That's why a assured nutrition is provided to the embryo. As a result, this is an adaptation to provide assured nutrition to the developing embryo. Hence, endosperm development it precedes the embryo development. That is, the endosperm is formed first, and after that, the embryo is formed. Now, though the seeds they are greatly different, the early stages of the embryonic development in case of both monocotyledons and dicotyledons is similar. The figure which we will go through, it will show us the stages of embryo development in case of dicot embryo. Now, first the zygote will start dividing and it will give rise to pro-embryo, and later it gets converted into globular and heart shaped embryo which further develops into mature embryo. Now, this zygote it has been divided and forming the mature embryo. Now, a typical dicot embryo, if we see, it consists of embryonal axis as well as it consists of two cotyledons. Now, the portion of embryonal axis which is present above the level of cotyledon, that portion is known as epicotyle, while the portion of the embryonal axis below the level of cotyledon is known as hypocotyle. So, epicotyle it is the level above cotyledon and it terminates into plumule or stem tip which further develops into shoot, whereas the portion below the level of cotyledon is known as hypocotyle and hypocotyle terminates into radicle or root tip, and this root tip is also covered by a root cap so that the root tip can be protected while the root is growing. So now here in the diagram if you see the zygote it divides into two cells. These are the stages of the embryo development. After formation of two cells, zygote further keeps on dividing, resulting into the formation of globular embryo, as well as we can see that there is a formation of suspensor and at the top of the suspensor, you can see that there is a large cell which is known as hostorium. This globular embryo, it further develops to give rise to a heart-shaped embryo, where we can see that already this heart shape is formed because of the development of the cotyledons, and this development of the cotyledons, it further develops and matures, giving rise to the mature embryo, where we can see that both the cotyledons have developed well, and there is a plumule which is present above the embryonal axis, and radicle is present below the embryonal axis. So here the portion above the embryonal axis is known as epicotyle, whereas the portion below the embryonal axis is known as hypocotyle. So this epicotyle will terminate into plumule, and hypocotyle will terminate into radicle and root tip, which is further covered by a root cap. Now, if you see the diagram A and diagram B, the diagram A is a dicot embryo, where we can see that there are two cotyledons. There is hypocotyle, radicle, root cap, and there is a plumule which is present. Whereas on the other side, you can see that there is a diagram of embryo of grass, which is a monocotyledon, where we can see that there is only one cotyledon which is present towards the side. Along with that, it consists of shoot apex, radicle, and root cap. Shoot apex is covered by coleoptile, and the radicle is covered by coleorhiza. Now moving towards the next topic, that is development of embryo, which takes place in case of monocotyledon. Now as we have gone earlier that the initial stages of the development of the embryo in both monocotyledons and dicotyledons are same. Embryo of monocotyledons, the difference is it possesses only one cotyledon, whereas in case of dicotyledons, they have two cotyledons. Now in the grass family, the cotyledon is known as scutellum, and it is situated towards one side of the embryonal axis. Now if you see the diagram which has been shown here, the scutellum, which is the cotyledon of monocot plants, it is situated towards the side. On the another side, we can see that there is an epiblast. Now this epiblast, it is basically considered the second cotyledon which is present in monocots, but it is not being developed. As a result, this cotyledon remains undeveloped. That's why it is also known as second cotyledon which is not been developed, and scutellum is the cotyledon which has been developed. So in monocots, only the scutellum will develop. As a result, only one cotyledon has been developed. Now, as the scutellum is situated on the lateral side of the embryonal axis, this lower part or the lower end of the embryonal axis, we can see that there is presence of radicle and root cap, and on the upper portion of the embryonal axis, that is above the level of attachment of the scutellum, there is presence of epicotyle. Now, radicle and root cap, they are covered by an undifferentiated sheath which is known as coleorhiza, that is for the protection, and the epicotyle, which is present above the level of attachment of scutellum, it has few leaf primordia, and it is enclosed in a hollow foliar structure, that is coleoptile. So there will be presence of few shoot apex, and this shoot apex is covered by coleoptile. So basically this coleoptile and coleorhiza are the covering sheets for the shoot apex and the radicle in case of monocotyledons, and they are protective in function. So in the next class we will go with the development of the seed. Thank you very much.